Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello, everybody. Welcome back to another episode of the A-Level Cookbook. Today we're going to be doing the OCR A-Level Biology A 2021 Paper 3. The figure below shows a light micrograph of an insect's gas exchange system. So how it works in an insect is they've got this opening in the surface called the spiracle, and then they have this tube here like this. That's called your trachea or tracheae because they have multiple of them, and they'll branch off into tracheoles and stuff. So we have a look here. We've got that sort of look here, right? And they all end in this bit here. To me, that means A is a spiracle and B is a trachea, not tracheole because it's not all the branchy bits. So there you go. Spiracle and trachea. Fish use gills as a specialized gas exchange surface. In ventilation, water moves into the buccal cavity. So buccal just means mouth, and cavity hole, so mouth hole. Across the gills and out of the opercular cavity. Complete the table by placing ticks in the appropriate boxes to show which of these processes occur at each stage of ventilation. Fine. So if we draw my epic drawing of this fish, right? Look at that. It's got sunglasses too. He's so cool. Look at that. This is your buccal cavity, the mouth. These holes-ish, these flaps that are open, is your operculum. So what happens is that these geezers open their mouth, water gets in, and what they'll do is they will then close their mouth, right? And then it will shove the water through this way like that, over the gills and out of the operculum. The operculum is just a bony flap that protects the gills, but when they close their mouth, it kind of opens those opercula on either side. The reason being is because of changes in pressure, right? So in their mouth, when it's open, the water's nice and happy. Imagine we've got like 4 of them, right? Chilling. Then when they close the mouth, the volume of the things, the volume of the thing that the water's in is a lot smaller. Can you see all these particles are all closer together? The pressure's higher. So if you think of it almost like this box here with a pipe like sticking out, right? If they then close the mouth, that box is a lot narrower, so it shoves the water that way. In the same way, when you get a water bottle, right? Imagine that's a water bottle, and you squeeze it, the water comes out the top, doesn't it? It just comes out the top. The reason is because the volume in here is lower, so the pressure goes higher, and outside it's a lot, the atmosphere, the pressure's a lot lower. So things like to move down a pressure gradient. So what they're doing here is essentially putting the water in their mouth and then closing their mouth, forcing it to go that way, because that's the route of least pressure. It's back out into a happy place. So that's what we're going to do. So we need to describe all of those. So here then, it's saying put the ticks in the boxes which show which process happens at each stage. So in this case, water moves into the buccal cavity. That's when they open the mouth. Now the buccal cavity floor just means the bottom of their mouth, right? So that's going to be that, right? Then none of the other ones really, because if the operculum opens, it's going that way. It's getting out of the buccal cavity. And also highest rate of diffusion in the blood, but this is just in the mouth, not in the gills, where it's just all kind of like behind there. So then that's that row done. So then here, mouth closes, water moves across the gills and out the opercular cavity. Well, yeah, because if they do that, it forces the water over the gills and out that way. So that's true. Buccal floor cavity floor lowers. No, it doesn't. It goes up because that's how they close their mouth. So it's not that. The operculum opens. That's true again to let the water back out. And the highest rate of diffusion in the blood, yes, because it's passing over the gills. So that's why it's those. There you go. So a student observed how they dissected a fish to view gills. I held the fish on a cutting board with one hand. I used scissors and scalpel to carefully cut the mouth from the tail down to the ventral side of the fish. I was able to split the fish into 2 halves and view the gills on the side and the inside of the mouth. So just one improvement the student could use to allow them to observe the gills more easily. So cutting a fish from the ventral side means you're cutting it from below. So imagine that's like you looking at this fish. I know my drawings are so cool. From the bottom, right? They've cut it like that from the bottom. Now there's some other things you can do to make it more easily visible. One thing you can do is that remember that these operculum, if I can find a diagram. Okay, this is going to be a really tough drawing to do, but let's say this is our fish chilling, right? Let me think, how do I want to draw this? So what they've done is they've cut below and then they've sort of viewed it as in like looking at the inside half, right? Like this. So it's like a half of it and they're looking there. One thing that could be easier is if they just took the fish, right? and then just cut out the operculum and moved out of the way. Because if I can find a picture, one sec. Okay, I can't really find a picture, but let me just doodle it because it's hard to explain without just drawing a picture. So here's the fish. And like, if I move, imagine that's like a flap of operculum. If I move out of the way on the outside, you can see the gills really nicely here. Ordinarily, that's kind of covering it because, again, it's the whole point of the operculum is to cover it. So this person, rather than just chopping it and seeing the inside half, could just remove the operculum. And that'd probably give a much better view, really. And alternatively as well, you shouldn't really be holding it down one hand like that. That's not very safe. So they could put something to hold it down, like pins or something, to make sure that it stays in place. But that's what I would go for. Just remove the operculum. That's like the more obvious thing. So lungs are specialized gas exchange surfaces in mammals. Dogs are mammals. A disease called canine pulmonary fibrosis can affect lung function in dogs. CPF can reduce the tidal volume of a dog's lungs. The West Highland Terrier developed CPF more than any other breed of dog. The lung function of a West Highland Terrier was tested. At rest, its ventilation rate was 1.44 decimeters cubed per minute. So 1 minute had a rate, ventilated this much. And its breathing rate was 24 breaths in a minute. Calculate the tidal volume in centimeters cubed. So the tidal volume is the volume of air with each breath, right? So if we have 1.44 decimeters cubed moved in total per minute, right? And we're taking 24 breaths in 1 minute, then that means 24 breaths moves 1.44 decimeters cubed. So therefore, one breath would be 1.44 divided by 24, which if we do that, we get 0.06 decimeters cubed, but they want in centimeters cubed, so you times it by 1,000 to convert that. So then you end up getting 60 centimeters cubed. Explain how the high occurrence of CPF in West Highland Terriers could have been a result of artificial selection. So this is also known as selective breeding, right? If I have like bananas, right, and I want them to have lots and lots of fruit and barely any seed, because original bananas used to have chunky seeds in them like that, and we didn't like that. One of the times, randomly a mutation will have happened, giving us a nice banana with teeny seeds. And we were like, yo, I want more of that. We were like, yo, I want more of that one. So then we end up letting this one, breed, however banana plants breed, right? Until eventually barely any of these are left behind and we had loads of these bananas with this stuff, right? That is selective breeding. The selection pressure, if you will, is us saying, yeah, we want this one, you're not breeding. The entirety of selection hinges on the fact that either you are able to reproduce or you're not. Everyone says the survive part. That is true. You've got to survive up to the point of reproduction. But if you don't reproduce, you're not passing your alleles on, right? Here, this one's not passing its seed alleles on because we're saying to it, nope, you're not allowed to breed, you're not allowed to reproduce. But you can, right? In the same way here, this is what this question is referring to because that's artificial selection in this sense too. We need to figure out why artificial selection in this dog breed would be an issue. Well, if you think about it, with dog breeds, we have certain traits that we like about them. Some people like the absolute hideous looks of pugs. I think they're actually just really ugly. No offense. SARS. Other people are like, oh my God, it's so cute, right? That's because we've bred them to be like that and we keep breeding them like that because, humans unfortunately just like them looking like that. And also there's that big fat profit cash payout as well. So in this sense, what's going on is that because we've been doing that, in that banana example before, all of these bananas now are nearly identical in terms of their alleles and their makeup. There's very little genetic diversity. Now, the problem in having little genetic diversity is that the risk of things like, one disease wiping all of this stuff out is like higher because they're not, they don't have like variable, different alleles and some that being more linked, like more likely to survive in this and that, blah, blah, blah. But in the same way, because we're also reducing the genetic diversity, things like recessive alleles are more likely to be, present. They're more likely to be expressed because you're now making these things inbreed with each other and you've got a smaller gene pool, smaller smaller supply of alleles. So in this case, what they must have done is that this selective breeding for desirable traits has reduced genetic diversity. And now using the context of the question, it's a disease called this, but it's not telling you it's like infectious or anything. So what most likely happened is there's probably like some sort of allele that's causing this disease. So because there's less genetic diversity, there may be a recessive allele for CPF, which is more likely to be expressed now because there's not as many, dominant alleles to mask its expression. There you go. Explain how DNA sequencing can help scientists understand how the West Highland Terrier's genes affect its probability of developing CPF. So we can use a mixture of this and bioinformatics because DNA sequencing, right, is all about just, working out, identifying an organism's genome. The entire thing from every single base. Right? We can get a lot of different sequences from these, and then what we can do is we can use bioinformatics. Bioinformatics is where you're using software to store, analyze, and do statistical tests on, biological data. And some of the uses for it are genotype-phenotype relationships. So what this means is that they can sequence a whole bunch of dogs and then look at their phenotypes and see what's up, as in compare them. So by comparing them, they might find patterns amongst all of them and be like, ooh, this could be correlated, as in it might be linked to the fact that they have CPF, right? So you can use that to identify possible alleles that can cause it and then use that to sort of figure out the risk of them developing it, because that's another thing that you can use bioinformatics for is epidemiological studies. What's more likely that's all about the study of distribution of a disease and its risks and stuff within a population. So if you have this allele, you're more likely to have this condition, that kind of thing. So in this case, what we can do is exactly that using these two. we can use sequencing to compare genomes of dogs with CPF, right? Well, this one, right? Against these dogs without CPF and see, we could see if there's a specific allele for CPF, right? You can also use bioinformatics to see if any specific base sequences are linked to having CPF, and that'll help you work out the risk. So another disease that affects dogs is caused by parvovirus. Dogs can be vaccinated against parvovirus at 6 weeks of age. Suggest what it's likely to contain. Whenever you want a vaccine, the entire point is you want to inject an antigen, because an antigen, foreign antigen, is the key thing that will stimulate an immune response. So foreign antigen stimulates an immune response. Not just pathogen, this and that, blah, 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 because it's the foreign antigen on the pathogen, or a toxin acting as a foreign antigen, or a cancerous cell that has mutations changing the proteins in it that act as foreign antigens. That's what stimulates the immune response, right? So when we're using a vaccine, we're trying to tactically put an antigen in to stimulate an immune response again while also not making them sick. So what you can do is you could either use, antigens from the virus, or you could use a weakened version or dead or inactivated version of the virus. Or you could, like the COVID vaccine, put its mRNA in so it makes the virus proteins in the same way we make the spike protein with the COVID mRNA vaccines, right? You can't say dormant form of a virus because once it gets in, that's you infecting them with it, not actually giving them just the antigen. So dogs need a booster vaccination against parvovirus when they're 1 year old. Explain why a booster vaccination is required. So the entire point is to make memory cells. That's it. The idea is that by giving them that first immune response, they'll have their memory cells and whatever. And then the second time you'll do it, you'll get those big spikes in antibodies and stuff like that and all this lovely stuff, right? But you also get more memory cells as well. So you do that to make sure that those memory cells are produced and also persist. So that's probably why as well. So it's to ensure memory cells persist in numbers. That's all. It's the same way, for example, with the vaccines that children have, like the MMR and, all of those kinds of ones, right? They have repeated doses to make sure those memory cells are up there. So now it says, describe the events taking place in cells D, E, and D. So if we have a look at cell C, then what do we see in here? So this is about undergoing mitosis, so we're going to look at the clues in the picture. So rather than look at it and then try and figure out, go in thinking what you're looking for, right? Each stage has its own unique characteristics. So prophase, you've got no nucleus anymore. The nuclear envelope is gone. The chromosomes are lying free in the cytoplasm, and you can actually see them now rather than it just being like a jumble of stringy stuff that you actually can't see with a microscope, right? Metaphase, middle phase, they're all going to be in the middle. Anaphase is when they start getting pulled and they look V-shaped as well. And then telophase is where the chromatids are in opposite poles and the cell starts to divide with cytokinesis, right? So you're looking for clues like that. So go in thinking, all right, I'm going to expect to find this for each stage. If I look here, I can't see a nucleus. So immediately we've at least gone prophase or further, right? But these aren't nicely in the middle and they're all sort of visible. So you can actually see them now. So it's not reached metaphase yet. So it's got to be prophase. So we need to explain why. We can't just say, describe. We need to show what the evidence is saying here as well. So it's prophase because, well, one, the nuclear envelope has disappeared because I can't see it. They're all just chilling in the cytoplasm, right? The chromosomes are visible now, and that's because they will have condensed and shortened, right? Oh, sorry, and prophase is C. Now, if you look at, there's nothing else really I can add to this, to be honest. That's all we can see. So next we're going to look at D. Can you see how there's these things on the chromosomes yanking them? Those are the spindle fibers, right? And they're going to be on opposite poles, but can you also see how they're in the middle and they're starting to look V-shaped? So with prophase, we've already gone past that. In metaphase, they'd be in the middle chilling without these actually becoming V-shaped yet. They would kind of look like this, right? Which it doesn't look like here. So I think we're getting past metaphase as well, but we've not exactly gone to telophase point yet because there's not, lots of different ones in each side. They're not on each pole very clearly yet, and there's no cytokinesis either. So in this case, this has got to be anaphase. So anaphase is in D. Why? Because you can see the spindle fibers are contracting. That's what those lines pulling on them are. So the spindle fibers are contracting and they're pulling the chromatids, because remember that's what's happening in mitosis. At this point, the chromatids separate, because in mitosis, what happens is you've got your here, right? So normally you're chilling, you're diploid, maternal, paternal, right? Then you have maternal, paternal, because you've copied everything. These are homologous chromosomes. These are also homologous chromosomes. They don't separate because we want to be diploid at the end. What ends up happening is these chromatids separate in the cell. So you end up with a maternal and a paternal, separate ones, they're not joined, and then a maternal and a paternal in the two daughter cells like that. So we're pulling the chromatids apart in this. We're not pulling, we're not separating the homologous chromosomes. So here we're pulling the chromatids and they are V-shaped. There you go. I should really say to other opposite ends of the cell, really, because that's what we need to say as well, because that's the direction they're headed in. There. So now if we have a look at E, this one, you can see the cells dividing and you can see a new membrane starting to form there. And as well as that, the chromatids are on either side of the cell. So we're now at really late end of the stage, telophase, right? And cytokinesis, because in telophase, they move to the opposite ends, the spindle's uncoiling, and they're starting to be all happy and gone, and then the nuclear envelope starts to reform. So we're at that sort of ND point there. We've got two nuclei forming, and then cytokinesis. So E is telophase. If we want to be specific, late telophase. Why? It's because the chromatids are on opposite ends of the cells, and also there's a new membrane forming in the middle, which is indicating that cytokinesis is happening. So you need to, when it says describe the events, don't just simply write down what the stages of mitosis are and all that kind of stuff. You need to say what you're seeing in this diagram and why. I'm not seeing all the other bits and bobs very clearly. I'm not seeing that a new nucleus is, forming between each of them. So I'm not going to yap about it and lie, Like, you've got to say what you see in the diagram. So the eukaryotic cell cycle is regulated by 3 checkpoints. Mutations can occur in genes that control the cell cycle checkpoints. Scientists recorded observations of 2 different tissues. In one tissue, they found a genetic mutation that stopped the metaphase checkpoint from working, suggests an abnormality the scientists might observe in the cells of this tissue. So we have some multiple checkpoints. The checkpoints that we have are your G1 checkpoint, the G2 checkpoint, and you also have the metaphase checkpoint. It's one of these. So in the G1 checkpoint, what happens is the cell essentially checks for things that are needed to prepare for S phase, right? So we're checking, is there any damage to DNA? And also, do we have what we need to do DNA replication? That's that one, because we haven't hit S yet. Gap phase 2, the checkpoint here, is to check if all the DNA has been replicated, because it's after S, right? So if it's now replicated without any problems or damage, then it's able to go into mitosis, right? But then we also have the metaphase checkpoint. The metaphase checkpoint will check that all of the chromosomes in metaphase are attached to the spindle fibers, like this, right? Here and here, blah, 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 like that. If they're attached, then mitosis can continue. So this question's told you that there's a mutation that's stopping the metaphase checkpoint from working, right? But the cells have still been able to, get to this point so far. So what's happened is if, we aren't having the right amount of them attaching to the spindles, they'll have the wrong number of chromosomes. So there you go. Cells have incorrect chromosome numbers, because if you think about it, if this checkpoint's not working, it's just going to let it carry on even though some haven't attached. So what you end up with are cells with varying numbers. If these ones don't attach, for example, some might have more and less. In the other tissue, the scientist observed cells and chromosomes that had replicated despite containing damaged DNA. Which cell cycle checkpoint is no longer working? And justify your answer. So here it's told you these tissues are replicated, but they still have damaged DNA, right? So if we think about it, the DNA has been replicated, number one, right? So it must have been that the damage was present beforehand, because if it was damage that was due to the replication not working, then you would have a G2 checkpoint problem. So the fact that they're able to replicate is because they were able to get to S, and once they've done S, it's just checking whether it's replicated successfully here, if that makes sense. It's not, the G2 one can't really check very much else. So G1 checks that the DNA you've got is fine to move on to S that replicates, whereas G2 checks the replication was fine. So here, it's got to be the G1 checkpoint, because the questions told you that they had replicated despite containing damaged DNA. They're going to replicate the DNA is damaged. It's just the G1 checks if it's damaged first to stop it from getting there. So G1 checkpoint. Why? It's because the damaged DNA should have stopped the cells entering the S phase. This is the point where you're checking for DNA damage directly like that. The G2 phase checks to see if the DNA has been replicated without damage. You can go in with damaged DNA, and as long as it's been replicated fine, it might satisfy G2. It's G1 that's stopping that. So mitosis does not occur in bacteria. Bacterial cell walls divide by binary fission. Binary fission is shown in this figure. So bacteria do not have a nucleus. Give 2 other differences in which the DNA is separated during binary fission and describe mitosis. So here then, can you see how there's just a loop of DNA, loop of DNA, loop of DNA? It's not, condensing into those nice chromosomes. So no condensing of DNA before, separation. As well as that, we have replication of plasmids, don't we, as well? And they can be in varying numbers as well. But here it's in the same, so we're going to write that. Other things you could say as well is that there's no, metaphase bit. They don't line up in the middle, and there's no spindle fibers, and there's no, separation of chromatids. It's literally just loop of DNA, and a bit more plasmids, split. There you go. So scientists recorded the population growth of bacteria in a closed culture. The scientists added various nutrients, including nitrate. So figure 2.3 shows the growth curve of this bacterial population. So if we have a look, we need to figure out what our independent and our dependent is, right, in this sense. So independent and dependent, we're looking at growth of bacteria. So obviously time is changing, but we're also changing the nitrate 10O3 minus concentration, right? And as well as that, our dependent is the number of cells and bacterial cells, right? So we're looking at the effects of different nutrients, in this case nitrate for this graph. Now this is a very nasty graph to interpret, but if you do it step by step, it's not too shabby. What they're doing is they're looking at how things are changing with time. So as time goes on, pick a graph and do it one by one. Bacterial cells are circles, so you're going to use this axis, right? So as time goes on, the bacterial cells go up, and then a bit down, and then up, and then they go up and up and up and up like that, right? And then with nitrate, at start, nitrate was like, what, minus whatever this number was here, right? And then it's going down, 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 down as time goes on. And as it's going down, the bacterial cells seem to be going up a little bit dip, and then they're also happy and up. And, at the time where we're hitting like, day 5 or whatever, there's no nitrates left. So you got to do it line by line, one by one first. Can you see how I did this first and then this? And then I compared both to see if there's a pattern between them. If you do that, then you won't get lost in the graphs. So the student looked at this figure and made this statement: the bacterial growth curve looks very different from a standard growth curve for bacterial populations. Use evidence from this to evaluate the statement. So whenever you're evaluating, you're looking at supportive statements, you're looking at contradicting statements, and then you're looking at issues with validity. So what I mean by that, and I've said this in every other video really, you'll see that I've used the same example. Supportive statements are things that are supporting them, as in like what in this data shows exactly what this person is saying. This person is saying that this figure is very different from a standard growth curve, right? So every point that's for it, all right? And then you need to show why. Contradicting is what in this data shows and directly against this. What actually says, no, it's actually similar. And then issues with validity is, does this answer the original research question? Can I apply this to every single other scenario? Will I get the same results every time? Is it repeatable, reproducible? Has everything been controlled? Have we got enough information to make a conclusion about this? Have we got a statistical test? If I give 10 people in a room chocolate and they all shit themselves, 100% shit rate, right? And then I say, oh, well, everyone who eats chocolate must be lactose intolerant and also will shit themselves. Then you can't go and apply that to everyone else, because if you got 10 people in a room and gave them chocolate, not all of them might shit themselves. So a supportive statement in that scenario would be, yes, I guess 100% of your sample did shit themselves. There is no contradicting thing there. Whereas if I had 9 out of 10, then you could say, well, one of them didn't shit themselves, right? But, there's no contradicting statement. The validity issues are all to do with what's wrong with my experiment. The issues with validity include, well, I've only done 10 people, there's small sample size. I don't know how I selected those 10 people. What if they had allergies? Or what if they just didn't like chocolate? And what if they had an illness at the same time? There's lots of reasons why, right? And then as well as that, I didn't know their age, sex, and health. I didn't know the chocolate that they were given, how much, how little, how much lactose in the chocolate, whether the chocolate had any lactose, whether it was, like, what else did they eat during the day? How long did I wait to see how long they'd shit themselves? Maybe it'd been months and years, and then all of a sudden they shit themselves, and I'm like, yay, woohoo. And no statistical tests. What even would you do in a statistical test? In that scenario, you could do like a chi-squared, for example, observed versus expected, but that'd be a bit useless in that scenario, because again, what would you do, And as well as that, there's no control group. The control group the study here is chocolate shit themselves. So I need a control group where I'm giving them nothing or no chocolate to make sure that if they shit themselves, it's because of the chocolate. A lot of people also like to say control groups are allowed for comparison. You're going to compare every single group anyway, so it doesn't tell us why. You're going to compare, if I had like 3 different groups, I'm going to compare all of them. That's the whole point of having different groups. The reason you have a control is to make sure that you're independent, the thing you are changing is the only thing affecting the dependent, the thing that's measured. So in my shitting example, my control group would have everything, the same thing, except no chocolate, or maybe like chocolate with no lactose in. So then I know for sure that if anyone does shit themselves, as long as I've controlled everything else, the only factor changing between these two things is the chocolate or lactose. So I know that they're shitting themselves. That's what the whole thing to do with validity and this kind of stuff is. So with here, the supportive statements, you should also try and back them up with evidence from the graph and also possible explanations as to why. So if we have a look then, your standard growth curves have the sort of lag phase here, your exponential phase, your stationary phase, and a decline phase. Now here, there's no clear lag phase, is there? They just seem to go up, then down, and then back up again, right? So that's already a pretty good, supportive statement to say that this is very different from our usual curve. And as well as that, there's not really a clear stationary phase or decline phase either. So then you should put some numbers to it. So if we have a look here, from days 3 to 4 to 5, or from the start, honestly, there's no clear lag phase. And also, why is it going up and then down? So there's a dip from about 3 to 5, which is a bit odd. So supportive statements is there's no clear lag phase, and also stationary phase either, or death, to be honest, or decline phase. As well as that, the dip at days 3 to 5 is not usually seen, because you don't see a dip, do you? You just kind of see a lag and then they grow. Now, that's the supportive statements. The contradicting statements then, well, here there is a bit of a stationary phase. We just might not have had enough time to actually get to that decline phase yet. So stationary phase might be present, it's just not enough time has elapsed. Because if you left it for a bit longer, maybe they do start declining right after, right? What else is contradicting? So what else makes this the exact same? Well, if you have a look here between these days, you still have an exponential phase, don't you? Even though there's no lag phase, there's at least still that. So again, you're looking for anything that's contradicting, as in matching the opposite here. So there is still an exponential increase at days 1 to 2. Now here, this is a bit hard to talk about because here we're just looking at scientists doing this and the population growth in a closed culture, and they've done various nutrients, including nitrates, right? Like, it's kind of hard to do our validity statements because we don't really need to talk about a statistical test here and this and that, blah, 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 because they're not trying to draw a conclusion like, oh, if you give them nitrates, they'll grow faster. Here, it's just talking about what makes this different to a normal curve. But the points I make for the other stuff was more relevant to like when somebody says, oh, if we give bacteria nitrates, they'll grow amazingly, kind of, if that makes sense. Because we're comparing just this graph being different from another one. It doesn't really matter about like, statistical test and this and that, blah, blah, blah. Now, we can think about a possible validity issue. If we look at the pattern of what's going on here, bang, it just shoots down like that. And then it just kind of levels off here. So what if they just needed some more nitrate? What if we gave them a nice fatty supply and then they went bang like that off the thing? So that could be a possible like sort of validity-ish issue, is that it could be they just needed more, needed more nitrate. Maybe this, created this non-normal curve. But the other points still stand. If you have another sort of thing where it's like, oh, well, a scientist looked at this and then concluded this, then obviously you can start bringing in the things about the, p-value bit and that and bits and bobs. So it says, calculate the total number of bacterial cells that will have been present in a 50 centimeter container on day 1, write your answer in standard form. So we're looking at day 1. If I draw a line here, we're looking for where this, remember, is the nitrate, right? So be careful, don't get thrown off by that. We're looking for the bacterial cells, which is here. Now that's about 4.7, right? If you look at the units, it's a log 10 number of cells. So the log 10 value is going to be 4.7, right? And this is per centimeter cubed. That's what the CM minus 3 means. So if I have 1 centimeter cubed of something, I'll have 4.7 log 10 of whatever this mysterious value is. To undo a log, all you do is you just do the base, which is here, 10 to whatever the value was. So 10 to the 4.7 would tell you the number of cells per centimeter cubed, which means in 1 centimeter cubed, I'll have this many cells. So if I want 50 centimeters cubed, I would do 50 times 10 to the 4.7. If I do that, we get 2.506 times 10 to the 6. Should try and stick to the number of significant figures they gave us in the question, which is 2. So we're going to go with 2.5 times 10 to the 6. There you go. Describe a laboratory procedure that the scientists might have used to estimate the population size. So what you're going to do is you would get, this is the experiment where you get, where you would put samples on an agar plate and grow and count the colonies, right? Now the issue is if I have bajillions of cells, if I put them all in a colony, I'm just going to end up with a dark thing like this, and it's going to be really hard, or like loads and loads and loads of cells. So what you want to do is you want to somehow make the numbers appear smaller here and then be able to scale them back up. So what you do is you dilute the original sample first. If you dilute the original sample, for example, if you do like, I don't know, a 1 in 1,000, then you know that if I count 1, 2, 3, 4, 5, I know that the original thing would have been 10,000 more, if that makes sense. So that's a helpful way of getting around the issue of trying to count like a bajillion cells. So what you would do is you would first of all do some serial dilutions and record them, obviously, and then you would grow colonies on an agar plate, count the colonies, and then you would scale up or undo the dilation, basically, to estimate the original population. That's quite commonly used. There you go. So the passage below shows some uses of microorganisms in biotechnology. Complete the passage by writing the most appropriate word in the blank spaces. So microorganisms have many characteristics that make them useful in biotechnology. These characteristics include something, life cycles, and low energy requirements. If you're growing things, for example, like you're making them, make insulin, we kind of want them to grow pretty dang fast. They don't want to wait ages for them to do it. So here it's going to be short lifestyle. Species such as Pseudomonas putida and Dechloromonas aromatica can be used for bioremediation to remove. So bioremediation, it's in the name, it's remediating using biological agents, as in we're using that to remediate something, as in fix something. So in this case, what we're doing is we're removing pollutants, wastes, contaminants from sites. So for example, things like using bacteria to clean up oil spills in the sea. That's an example. So that's what I would say is to remove pollutants. That's the whole point. Bioremediation's in the name. Other species of bacteria used to manufacture drugs such as insulin, antibiotics, which are secondary something, are produced by fungi and used to kill pathogenic bacteria. So organisms, when you're growing them, especially when we're doing in this context, they're going to produce things, secondary meaning not essentially needed for the direct growth of them, right? They're going to make things that are not directly involved in their normal growth, development, or reproduction. Okay? And examples of these are antibiotics from yeasts or like chemicals, types of chemicals that plants make, for example, to protect themselves. Those don't directly contribute to them growing. It just stops them from, suffering and competing with other things and bits and bobs like that, or maybe things like that. So these are called secondary metabolites because, again, they don't directly feed into it. Like a fungus making an antibiotic does not directly mean, it's going to grow. Like if you took the antibiotic out and assuming there were no bacteria there it's competing with, etc., it would just still grow fine. It would be chilling. That's a secondary metabolite. So here, that gap is metabolite. There you go. Produced by fungi and used to kill pathogenic bacteria. So there you go. So callose is a polysaccharide produced by plants. Callose is formed by beta-glucose monomers, and here's a section of callose. Here we go. Fine. So if we look at beta-glucose real quick, so there you go, and then you've got this, and you've got what, like this, there you go, then you've got CH2OH, and there's going to be an O there, sorry, not a bend, and then here we've got an O here, and OH, and this, and then a CH2OH, and then an OH, and then an H, and an OH, and then an H from the previous bit there. Is that all of the bits and bobs I need? There, right? It's very poorly drawn, very rushed, but here we go. If we look at it, says describe the differences between the structures of callose and cellulose. So already I can see there's a branch coming off of here. Cellulose is straight and unbranched. So immediately, callose is branched. Now, if we have a look at the other stuff, so we now have a look at cellulose. So in cellulose, remember that's your 1 carbon, that's your 2, that's your 3. So we've got a 1 here, right? This will be your 1, 2, 3, 4. So we have a 1,4 glycosidic bond, right? And this is in cellulose. So that's where your glycosidic bond is. So we've got a 1, 2, 3, 4, 5, right? So then here, that's 1. If we have a look here, that's going to be this one's 1 here. So 1, 2, 3. So you can see there's a 1,3 glycosidic bond here instead. So then it has 1,3 glycosidic bonds. So there's another one. When you're looking for sugars and monosaccharides and you're looking for their polymers, the glycosidic bond is this O here. It's either up or down depending whether you have alpha or beta, right? Or what else have they told us then? Some other things as well. Can you see how this cellulose, this is up, this is up, this is down. Can you see they're rotated? Here, they're all in the same orientation. So all beta-glucose monomers are in the same orientation. So there you go. Those are the ones that I would say. Now it says, plants produce callose in response to pathogenic infection, injury, and changes in environmental temperature. Scientists wanted to investigate the effect of temperature on callose production. Outline a valid plan that could be used to investigate the effect of temperature on callose production, and you should include a suggestion on how you would measure callose production, right? So what you need to do is come into this and say, all right, I need to figure out what my independent and dependent variable is and come up with a decent plan. So start by identifying your independent and your dependents. So independent is clearly obviously going to be temperature. We're changing that, right? Dependent, don't just write callose production. That's useless. We need to figure out how we're actually going to measure that. So here it's a bit tricky, isn't it, to try and figure out how on earth you can see callose and measure it. You can't really do like a Benedict's test for sugars or starch because, it's not starch. And also it's not going to be, callose is a polymer of it. So you're not going to see it with Benedict's test, right? You're not going to get any useful data or results. And in the exam report, it also says that a lot of people overthought this and tried to just jump into saying that. Here, you've got to come up with just something that would make sense. You've got to basically, chef up an idea. Now, if we have a think about it, what does callose actually do? So callose is deposited in cell walls and between cell membranes and stuff, right, of infected cells. And what it does is it acts as a barrier, right? What we could do is we can take tissue samples and look at it under a microscope, but it is callose and it's pretty small, right? Now, if we are looking at something that's small and not easily visible amongst everything else, and we're looking at a light microscope, with all the other tissues in the world that we look at with blood and this and that, we add a stain. They're not expecting you to know a specific stain. It's just they're trying to get you to test your skills and whether you can try and at least formulate some sort of plausible idea. And to me, if I'm looking at something under a microscope, with nearly everything we do, we look at it under a microscope with a stain because we're looking for something, right? Now, because this is also a bit more unclear and it's a bit harder to see, for example, it wouldn't be as clear as here's a cell, here's a no cell, right? It's a kind of, we're looking at a weird sugar thing. Another thing you can also do is immunofluorescence as well. So what you do is you use antibodies with fluorescent tags on them, and then you put it on a sample, and it will bind to the callose, and then you can look at how much is fluorescing, for example, under UV light. So this is a bit tricky. So what I think I'm going to do is I'm going to so dependent is what you're measuring, right? We need to find out a way to measure the staining. So I'm going to go with staining. I'm going to see how we can measure this. So we could put so I'm going to write the amount of staining of tissues. We'll come back to how we'll do this in a sec, right? So when you're setting up an experiment, the idea is because you've now established what your independent and your dependent variables are, you know exactly what you need to control because every single step in this should hinge on this being the only thing that changes this. So what you do is you want to select, a decent sample size of this plant, right? So select, 30 identical plants So like as in species wise and all of that kind of stuff, right? Ideally, if there's clones, and also other things to control as well are things like, for example, control their age, species as well, and size, right? Then what I want to do with these is I want to put them in different temperature environments, but we want to check the temperature in different intervals. So place in controlled temperature ranges, right? So like do it in increments of 10. So 10, 20, 30 degrees, 40 degrees, 50 degrees Celsius, right? And then what we do, so we've got a good enough sample size. We place them in a nice environment. And then what we try before the experiment, we need to sort of figure out a standard almost. So you know how you make your standard curve, right? In this sense, we need to try and figure out a way of making like a reference point, all right? So here we're looking at them under a microscope to see how much staining there is. What you could do is you could get samples of like known callus and staining and and produce a baseline. So what you could do is you could get like normal, chilling healthy plants in each temperature and see if they're higher or lower, right? So what you could do is you could get plants at standard temperature, like, room temperature and get an image of the amount of staining there, because then that can be a reference point. Alternatively, you could make a scale if you want, or something like that. But here you've got this picture that's sort of like, I don't know, whatever color it might be. It's this amount of purple, right? It's either dark or dimmer. And we're going to compare what we do to see if it's darker or dimmer. As well as that, when we're doing this experiment, we also need to make some other control variables as well. So we need to make sure the pH is the same, the nutrients are the same. What else? Water, light. Can you see how I'm thinking about everything that's screwing up this thing? So water, light, use sterile equipment to avoid contamination, right? So those are the things that I'm saying here. Now we need to try and, again, measure this thing, right? And all I've done is just put them in the thing and let them do their job. So here, what we also need to do now is we've got to actually figure out how to investigate them under a microscope. So what we'll do is you're going to take tissue samples from the same place. So take tissue samples from the same site. You put a stain on them, so stain, and then look under, observe under a light microscope. So we're looking at the effect of temperature. This is a bit subjective, but what you could do is you could compare it to that standard that we got. So compare to the standard at room temperature, and see if it's gone darker or lighter in terms of the stain, indicating more or less cellulose callose. Alternatively, again, you can also use that scale as well. You can make your own scale up and say, all right, this is how much I know is definitely going to be here. Here's this, and so forth. But it's a pretty tricky one, this question. It's just testing whether you can actually, chef up an experiment of your own, if that makes sense. So what I always recommend is identify your independent and your dependent and think, how am I going to actually measure this? So I'm measuring the amount of callus in it, right? The only thing I can think of is putting stains in it. I can't do anything else, really. It's going to be hard to look at it because it's a sugar. It's not like a living cell. And then, so what I'm going to do is I'm going to look at the staining and I'm looking at the temperature and I need to think, how will the temperature affect the staining? It might go darker, it might go lighter. So I'll have my normal reference room temperature one and I'll just compare all of them. So whether it's darker or lighter, and then just describe the steps and think of your controls as anything that's screwing up your dependence. There you go. So callus production reduces the spread of pathogens in plants. The table below lists pathogens that infect plants together with the features of the pathogens and examples of the diseases they cause. Complete the table below by writing the correct answer in these empty boxes. So we have pathogens and has membrane-bound organelles. It has a cell wall. An example of it that's caused by that is black sigatoka. So this is it can't be a bacterium or prokaryote because it's got membrane-bound organelles. And this exact disease here is due to a fungus. Now here, let's have a look. We know it's causing ring rot. So ring rot is caused, this is just examples you need to know, unfortunately, is that it's caused by a bacterium. And it doesn't have membrane-bound organelles because it's a prokaryote, but it does have a cell wall, remember, because bacteria can. This is a virus. No, obviously it wouldn't have either of these two. And an example of it is the tobacco mosaic virus. There you go. Many plants have defensive responses to herbivores. Say one example of a response that plants have against herbivory. So the whole point is herbivores are things like scranum, right? Like scran plants. Plants can't just get up and run off, right? So they'll have defenses here to stop them from being eaten. So they can have physical defenses like thorns and spikes, spiny leaves, inedible bits and bobs, right? And they can also make chemical things as well, like poisons and toxins, like tannins, for example. What they will do is they'll bind to digestive enzymes in the saliva of things that are eating them and activate them. They can also make things like alkaloids as well, which, for example, screw up their metabolism and can cause killing in that sense as well. So here then, it's just one of them, to be honest. So I'm just going to go with saying that they produce tannins. There you go, which is a toxic thing. Many crop plants are eaten by herbivorous insects. Humans use insecticides to reduce the consumption of crop plants by insects. Explain why an insecticide might be less effective after being used for many years. So we need to think about what's going on. So here we're looking at insecticides. We're trying to kill insects. Now, for some reason, the questions told you that over time, for some reason, insects are able to, live despite this insecticide being the case. It's almost like antibiotic resistance, where, you know, you have your random mutation, one of the insects are now able to survive and reproduce and this and that, blah, 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 and they don't get killed. So here, it's because they have resistance. The insects become resistant just by pure chance. Do not say that the insecticide is causing a mutation. Mutations are random and spontaneous. It's not that. How all of this works is if I have antibiotics, for example, imagine that's non-resistant, these are resistant, right? So I've got loads of these non-resistant ones chilling here, right? Just because of pure chance, random mutations just happen. And some of them will be, you know, resistant just by pure chance. The moment I introduce those antibiotics, these all get wiped out. And then these ones didn't get wiped out because they just, again, by pure chance, were just resistant. So then 1, 2, 3, 4, 5, 6, 7, 8, they replicate and they're able to survive and reproduce. The antibiotic didn't make them have some mutation and this and that, blah, blah, blah. It's just here, they just pure luck, right? That's how they got that. So mutations are just random and it's just by pure chance. In most cases, when we use them, it actually does just wipe them all out. So that's what it is here. Some organisms use disaccharides called trehalose as a respiratory substrate. Trehalose has similar structure and very similar chemical properties to sucrose, suggest how you would test for the presence of trehalose. Now, with Benedict's tests, you're looking for reducing sugars, right? But some disaccharides can be reducing as well, but sucrose is not reducing. But the question, we don't know if it is or not. We've just known that it's similar. So what we do is we do, you know, Benedict's test first. So, you know, you would add Benedict's reagent and heat first to see if there is any reducing sugars. If there's no precipitate, then there's no reducing sugar there, right? So then what we can try to do to test for non-reducing sugars is we can add dilute hydrochloric acid, we can boil, right? And then what we can do is we can retest, and what will happen is you might get a precipitate forming as well, right? The reason is because when we do it with sucrose, doing this, having dilute HCl actually hydrolyzes it back into glucose and fructose, for example, in the context of sucrose. So this is a disaccharide. If we do this, it will break it down. It will hydrolyze it back into its original monomers, and you might get a precipitate there because those are reducing. There you go. So amino acids can be converted to other molecules and be used in respiration. Here's the formula of 5 amino acids that can be used in respiration. And it says, here's an outline of the link reaction and the Krebs cycle. So oxaloacetate combines with acetate from acetyl-CoA. CoA goes back away, disappears. We get citrate, and then we get alpha-ketoglutarate acid. Fine. So then it says here, the table below has some information about 3 amino acids. Use figure 1 and figure 4.1 and figure 4.2 in your own knowledge to draw a conclusion about which amino acid is being described. Write your conclusion and a justification for each conclusion in the table. So here we have information about an amino acid. It's converted to pyruvate with the fewest changes. Some amino acid, we need to justify why, and then converted to alpha-ketoglutarate acid, the fewest changes, and then the highest amino acid with the highest RQ. Fine. So if we have a look then, when we're converting to pyruvate, what are the properties of pyruvate? Pyruvate is a 3-carbon compound, and that's really it. Like there's not really much, else to know about it. So if we have a look, we're looking for whichever one's most common to pyruvate. That's going to require the fewest changes. All of these have amines and C-double bond OHs and stuff like that, right? So whichever one of these has 3 carbons is the closest. This has 3 carbons, so that's looking pretty good. That's got 4, so it's going to require decarboxylation, an extra step. This has got what, like 1, 2, 3, 4, 5. That's got 2, so we're going to have to add C on somehow. And that's got like, what, 1, 2, 3, 4. So the only one that it can be is alanine, just because it has the same number of carbon atoms. That's the only reason I can think of based on the knowledge that you're expected to have at A level. So alanine, it's because it has the same number of carbon atoms. So you really got to look at what they've told you in the question and compare it literally side by side. So here, converted to alpha-ketoglutarate acid with the most fewest changes. So again, if we look at alpha-ketoglutarate acid, 1, 2, 3, 4, 5. Based on the fact I used it there, I'm going to try again here, right? So that's 3, we'd have to add 2 on. That's 4, we'd have to add 1 on. This is 1, 2, 3, 4, 5. So we'd have to add 1 on. Sorry, we don't have to add any on, whoops. And then we have 1, 2 here, but then we also have 1, 2, 3, 4, 5, 6, right? So in that case, it's going to be glutamic acid for the exact same reason. So glutamic acid. Why? Because it's got 5 carbon atoms. It's the same number. Then it says the one with the highest respiratory quotient. So remember that the respiratory quotient is just your CO2 released over your O2 used, right? Now, some substrates used in respiration have specific respiratory quotients. So RQ, if you have lipids or triglycerides, they tend to be 0.7. Proteins and amino acids are 0.9. And carbohydrates are like 1. That's our reference point, right? So the reason that that's 1 is because if you do, for example, the respiration formula, it's C6H12O6, CO2 released, you get 6 CO2s, but you use 6 O2s in. So it's 1 to 1. That's why it's 1. So here, it's the one with the highest RQ. It's going to be the one that has the most, because it's that way, because for, like I said before, C6H12O6 plus 6 O2 gives you 6 CO2 plus 6 H2O, right? In this balanced equation, all of that carbon came from here, and all of the hydrogens came from here. And then, we had some oxygens already here, for example, and here. So to reduce this amount of oxygen needed, we need to max out the amount of oxygen here, because then we don't need to add as much, right? And if you have a look, that's your CO2 released and your CO2 used. So we need to make this lower and make some amount of this oxygen higher. So if you have a look at all of these, this has got 2 oxygens, that's got 4 oxygens, that's got 4 as well, that's got 2, and that's got 2 as well. So at least these ones are out, right? So we minimize the amount of oxygens needed. However, it's not only dependent on how many oxygens are here, it's also how many oxygens you have and how many carbons. Because obviously if I have more carbons to make CO2, I'm going to need more oxygen as well. So we need whatever's got the highest amount of oxygen and the least amount of carbons as well. In that case, if you compare these by these, it's got to be aspartic acid. And that's exactly why. So in aspartic acid, here it is, it's because it has the highest amount of O compared to other elements. There you go. Now it says outline the reactions that must occur to convert alpha-ketoglutaric acid to oxaloacetate in figure 4.2. So if we have a look here, this is 1, 2, 3, 4, 5. This is a 5-carbon compound, but oxaloacetate is 4-carbon. So immediately, one of the things that must happen is decarboxylation to shove out a CO2. Decarboxylation is removing that CO2. So decarboxylation is one of them, right? As well as that, when you're doing your Krebs cycle, unfortunately, it's just a big long chain of things. This 5-carbon compound that you guys normally learn eventually becomes that oxaloacetate again, and along the way we decarboxylate it. That's definitely one of them. And what happens is you also end up reducing NAD to get reduced NAD. You also get substrate level phosphorylation, so you get ADP and PI makes ATP, and then you also get FAD becoming reduced FAD. And then also right before the end, you then get NAD becoming reduced NAD as well. So then in that case, that's what all of those steps must be happening according to this diagram, which is a 5-carbon compound getting back there. So in this case, we've got decarboxylation. We are going to make, reduced NAD and reduced, red for short, FAD produced, as in we've dehydrogenated this or we've oxidized this. And then as well as that, we've also made ATP as well. There you go. Most ATP is produced in the mitochondria by chemiosmosis. Outline how ATP is produced in the mitochondria by chemiosmosis. So this stuff here then, right? If we have a think about what's going on real quick. So you have your reduced NAD and FAD. This is all in oxidative phosphorylation. It's in the name. Oxidative. So it's using the oxidation of reduced NAD and FAD. And then phosphorylation. Phosphorylation to phosphorylate ADP. Right? So if we've got reduced NAD and FAD, they're going to oxidize. If they oxidize, they're putting out that electron because they've lost it. Oxidation is losing electrons. What this does is it'll move down an electron transport chain like this. Now, all electron transport chains in this context reduce the amount of energy in that electron because the electron's too excited. It's like, I'm free! And what happens is it's gotten so little electron, eventually it gets picked up by an O2 and some H+, and you end up with water. That's why that's the final electron acceptor that everyone keeps saying, right? That's very similar to what happens in plants, which I'll talk about after. While it's moving down this electron transport chain and losing energy, energy just can't disappear. It's got to go into something else. So the energy that it loses is used to pump H+ into the intermembrane space in the mitochondria like this. So we have lots of H+s here. The reason is because conveniently we have a channel protein here that is ATP synthase. So now we've created a concentration gradient that's higher here, lower here, using the energy from this. So then these H+ will move through here via facilitated diffusion back down here, and it will drive ATP synthesis. Their movement literally just combines these two together like that to make ATP. That's why it's called oxidative phosphorylation, because we're using oxidation to phosphorylate ATP. In the same sense, just real quick, non-cyclic photophosphorylation, right? The words have meanings. They actually are relevant. Non-cyclic means that this electron is lost and passed on to NADP. This is in plants, right? It's no longer, it's not cyclic, it's not returned. Photo is using light, and phosphorylation is to, again, phosphorylate something. The whole point of the light-dependent reaction is to make ATP and ADP. Sorry, ATP and reduced NADP. So in that case, we're phosphorylating ADP to become ATP. And it's the exact same thing. So in photosystem 2, what happens is, think of it in terms of the journey of the electron one by one, right? Do it in steps. Photosystem 2, light shines on chlorophyll, electron is released because it's been excited. It has way too much energy, it's free. So what it does is it'll move down an electron transport chain, right? And it will, put it in its place, basically. It'll make it lose the energy as it moves down the electron transport chain. As it does that, it then redeposits in chlorophyll here, light shines on it here and excites it again. It's now so gassed, it's got to go somewhere. So what happens is NADP comes along and becomes reduced and takes it away. So here, the final electron acceptor is actually the NADP, not oxygen, Because here, can you see how we lost energy, blah, 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 moves and eventually gets taken up by O2 and H+. That's making the water that we get, right? And that's where oxygen is the final electron acceptor. Whereas here, it's the same process, just a bit, one extra jump really, and you end up getting NADP being the final electron acceptor. However, it won't go and sit in any of these things if it has too much energy, because electrons having too much energy are free to do whatever they want. They're free, they've escaped. So while it moves down the electron transport chain, again, same thing, it's losing energy, but energy has to go somewhere, so you end up pumping H+ ions across like this. You're setting up a concentration gradient, and would you look at that, there's an ATP synthase here as well that does the same thing. So it moves through down here, down a concentration gradient back here, and it drives ATP synthesis like that, ADP plus PI. But the other issue is, however, in this scenario, these reduced NADs and FADs will go off and do their job. Here, though, this chlorophyll's missing an electron. So what then happens is light shines on water, and it splits it into H+, an electron, which goes back here and replaces the original one, and O2, like that. There you go. That's why it's called non-cyclic, because that original electron we lost eventually ended up with NADP and went off to the Calvin cycle. The light photophosphorylation part, the photo bit is splitting this into H+, E- and H+ as well. And photo was the reason the electron actually got released to begin with, and then phosphorylation is to phosphorylate the ADP and PI. So light shines the electron, right, which is not returned, hence non-cyclic, and that electron is eventually used to phosphorylate ADP and PI. And that's exactly it. So if you think about it step by step, electron release, moved down the electron transport chain, gets excited again, passed on to NADP. Its energy is lost as it goes down there, so the energy has to go somewhere. It's used into pumping H+, it moves down ATP synthase, bang. And then to replace the missing electron, water split. All of these steps are very confusing because everyone tries to learn them all in one go in multiple steps. Rather than that, try to just do it step by step. Electrons journey first, then this, and this. So in the mitochondria, sorry, there was an entire side rant there. It's the same principle. So what happens is that as the electron moves down, the electron transport chain, obviously you write the whole thing, it loses energy. This energy is used to pump H+ ions from matrix into intermembrane space. And this creates an electrochemical concentration gradient as well. So electrochemical gradient. So then what happens is H+ moves down this gradient through ATP synthase, and this drives ADP plus PI makes ATP. And there you go, that's how it's done. So a person's respiratory quotient changes when they exercise. Figure 4.3 shows how the respiratory quotient changes with the power a person exerts during their exercise. Power measured in watts increases as the intensity of the exercise increases. If you have a look here, the power is getting higher and higher, and the respiratory quotient is changing. Fine. If you have a look at where the power is not too high, respiratory quotient is what, between like 0.8 and 0.9. 1, so it's 0.9 something, and then it goes a bit down, and comes back up, and you can start to see it overshoots. Here, the C, the point at which we have carbohydrates going even higher. So respiratory quotient, remember, is CO2 out over O2 used. That might be useful. So now it says, what can you conclude about respiration as 0 and this and this and this, right? So if you look at 0 exactly, it's above 0.9, but it's not exactly like 0.9 or 0.8, right? So it's more shifting towards using, remember that respiration is not just use one or the other. You're using mainly something or more of this or more of that. It's like proportions, right? So if we were on 0.9 exactly, then you would be using pretty much nearly, proteins and amino acids exclusively. But because we're not exactly at 0.9 or a bit above, we're actually using a mixture of the two, or maybe even just mainly carbohydrates, if that makes sense. So here, at this point, we're using mainly carbohydrates if we're using a mixture of the two of them, or respired. Now here, with 50 watts, if we have a look, at 50 watts, we are here now. So we're now treading into territory where we are at, 0.8 to 0.9, right? At this point, we are more close to 0.8 than we are to 1 and 0.9-ish. So what we're now looking at is we're now using more lipids and proteins, because lipids are 0.7, remember? And proteins, or you can say amino acids, and these are 0.9. So if our numbers between these two, then we're probably using a bit of each. Whereas here, we're more close to 1 than we are 0.9, so we're using mainly carbohydrates. It's kind of like where you are in between them that matters. Then here, for some reason, we've overshot, we've gone above 1, right? Now that means we're putting out way more CO2 than we are using. So we're using a lot less O2 and putting out more CO2. So the only explanation for that, right, must be that we're using less oxygen and we're putting out more CO2. So the only explanation for that is that there's got to be more anaerobic respiration. There's no other explanation to it. It's because if you have a high respiratory quotient, it means you must be short of oxygen. So we've not got enough to supply what we need. So we're having to use aerobic and anaerobic respiration, because it's not like you just switch over to the other, using both, right? So if we're doing that, we're shoving out, way more CO2 than we have O2 used up. There you go. So amylose is formed from the glucose molecules produced in photosynthesis. Table 1 shows these, table 5.1 shows these 3 statements, which may be true or false. So amylose is one of the components of starch. It's the non-branched component, right? And it's made from alpha glucose. So immediately it's a storage molecule. You don't want it to be soluble or else it just disappears. How are you storing it? So it's false. It's insoluble. It doesn't dissolve. It's not branched, right? The other one that's branched is amylopectin. And also it is formed by condensation reactions because you're joining those alpha glucoses together with condensation to form glycosidic bonds. Figure 5.1 shows the rate of photosynthesis varying with light intensity in two plants, a fern species this and maize this. So if we have a look, it says, what can you conclude about the habitat of this compared to that? So if we have a look, this is the light intensity, this is the rate of photosynthesis. So D. Antarctica seems to have a really high rate of photosynthesis up to about this light intensity, and then bang, it just shoots down. So it likes to have, some sort of like sweet spot light intensity before it gets too high, and it's like, nope. Whereas if you look at Z maize, it's just going up and up and up and up and up and up. And if you went further, it probably goes even higher. So that means that this has a preferentially lower light intensity than Z maize. Otherwise, why would it just stop photosynthesizing? So that means, according to that this, whatever it was, what was that? Antarctica thingamajiggy. So we would say the Antarctica blah, 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 is max photosynthesis is at a lower light intensity than Z maize, which is still increasing, right? So that means, if we're thinking about, we need to link this to the habitat. So this means its habitat prefers to have some sort of like, sweet spot, right? And then bang, it drops. So if we kept going, maybe this would get even higher. So it must be that this Antarctica thing prefers having a lower light intensity in its habitat than Z maize. Therefore, this D Antarctic blah, 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 probably, its habitat is probably darker than Z maize. That's the only explanation that I can think of, because why else would it want, if it was surely as bright as it is, it'd be higher. And then obviously, yeah. So this is the percentage of natural maximum, remember? So if you're at 100, that's 100% of the natural. But if they went further and artificially made it even brighter, maybe even this goes higher and higher and higher, right? So that's what it's trying to get at, is that its sweet spot must be a low temperature, because this theoretically could keep going. So water availability can affect the rate of photosynthesis. They investigate the effect of soil water content and the rate of photosynthesis in two plants, and their xerophite called this. They took measurements at 6 different sites for each species. They measured the water content of the soil and calculated the rate of photosynthesis for each site. So we need to figure out what our independence and our dependence are. So we're looking at the effect of soil water content and the rate of photosynthesis. So in a sense, we're obviously, first of all, changing the species. Now, in a sense, we're also kind of changing the water content, although that's what they're measuring, right, as well. They're using that to quantify the change here. And then here we have dependence. Our dependent is going to be the mean rate of photosynthesis. Fine. The reason we do this is because then anything else has to be controlled as best as possible. So then we need to look for anything that screws up this, it screws up this, it screws up this, it screws up this. So we have a look. The mean rate of photosynthesis, this is going down, and this also seems to be going like what? So it's kind of stable, and then it goes up, and then it goes down. Here, the water content here seems to go down overall. Here, it's kind of the same, goes down, down, down. So it's not really got like a little bit of an up blip and then a down. Then it says, here's some statistical values for RS, fine. And here it then says they investigated the relationship between the water content of the soils and mean rate of photosynthesis using the two plants. Using the values in table 5.2, calculate the Spearman's rank correlation coefficient for water content and the rate of photosynthesis in Z maize and use this formula. Fine. So here then what we've got to do is we've got to muck about with this formula. So right, this is your correlation coefficient. D is the difference in ranks, right? N is going to be the number of pairs of data. This sigma means the sum of, okay? So what we have to do is we need to rank the data from lowest to highest, right? So if we see here, it's for the correlation coefficient for water content and rate of photosynthesis in these, right? So what we need to do is we need to work out the ranks of both of these quantities for Z maize. So if I just move this over here to the side, or I get rid of it for now, right? I need to work out the rank of the water content and then the rank of the photosynthesis. So what you do is you assign them all a rank one by one, from 1 to, whatever the highest one is, right? If two things are in the same rank, as in they're both equal values, then you will give them a number between like whatever it is. So if they both appear as rank 6, for example, then you would do 6.5. So we need to rank all of these one by one. So if I rank the water, this is the lowest, then that, and so forth, right? So it's 1, 2, 3, 4, 5, 6. Fine. And then we need to rank this. So if I have a look here, that's your lowest. So that's 1, then it's 8, then that's 2, that's 3, that's 4, that's 5, and then that's 6. So the rank difference is the difference between these two, right? So it's going to be 0, 0, 0, 1, minus 1, 0. And then it's going to be d squared, because rank difference is d, right? So d squared is going to be 0, 0, 0, 1, 1. So then plugging into the formula, RS equals 1 minus 6 times the sum of d squared, remember, which is just 2. So it's going to be 6 times 2 all over the number of pairs n squared minus 1. So can you see how we have 1, 2, 3, 4, 5, 6? We have 6 of them. So there's going to be 6, open bracket, 6 squared minus 1. If you do that, you end up getting 0.943. There you go. So now it says use table 5.3 to decide what the students can conclude from the RS value. So we worked out 0.943. This n is our number of paired data, right? So we're looking at this column here, the 6. We want a p-value that's a 5%. So we're going to look in this column here, and we're looking for this row. Can you see that's 0.829? So that means if our value is higher than that 0.829, that means we are higher than that 5% threshold, okay? And actually, when we're at, because we got 0.943, we're actually at that 1% threshold. So then what you can say is that the, there is a significant, and because this is a positive number and it's closer to 1, because a negative correlation is minus 1, right? And a positive is plus 1, 0 is no correlation. So there's a significant positive correlation. So now the students calculated the RS value for the rate of photosynthesis and this, and they got 0.7414. Now, if we have a look here then, so the same number of, classes and stuff, right, paired data, sorry. So if we look for 0.714, that is below this. We're not at the threshold for 5. Actually, it's a 10%. So that means there is no significant positive correlation. So the positive correlation was probably due to chance because that p-value is testing this correlation. It's not, that's all it is. You can't just say the results are significant. You need to say the correlation is because it's testing the significance of the correlation. Photosynthesis can occur in organisms other than plants. These organisms have photosynthetic pigments or are adapted to their habitats. The cyanobacterium, this thing marina, lives in aquatic habitats in many aquatic plant species. Carrier, whatever, marina has a high concentration of chlorophyll D in its cells and a relatively low concentration of chlorophyll A in the absorption spectra as shown below. So what that means is here, if we look for example, chlorophyll D, it's absorbing all of these wavelengths, it's not absorbing all of them and this and that, right? And then for A, it's absorbing all of these wavelengths and it's absorbing this and this and this and whatever. So it's not absorbing those in the middle, sorry. So it says, suggest why a high concentration of chlorophyll D is an advantage for this, right? So we have a look at chlorophyll D. It's told you in the question that this is an aquatic habitat. It's an aquatic habitat that it lives in and it has a lot of D. If you have a look at chlorophyll D, it's absorbing this, it's not absorbing that, and it's, not absorbing all of this, right? So the range that it doesn't absorb seems to be a lot broader than what chlorophyll A does. Can you see how chlorophyll D is absorbing this when A doesn't absorb as much? It's absorbing this when A doesn't as much. And also here, can you see how that it's absorbing all of this when A doesn't as much? And also all of this, and you can see that A doesn't as much. It's absorbing all of these wavelengths that, A isn't really absorbing. So what might be going on in its habitat is that there's different wavelengths available than A, so it's absorbing those. So there's probably different wavelengths available in in its habitat. Therefore, it's absorbing more of these wavelengths. There you go. Diatoms are unicellular photosynthetic eukaryotes. Diatoms contain high concentrations of this pigment called whatever. And this is the chromatogram, and it's showing you the pigments X, Y, and Z. So there's your origin, there's your solvent front. It has an RF value of this. Identify the letter X, Y, or Z that does that. So if we have a look then, what you would do is that remember that your RF value is the distance of the spot over the solvent front, right? So then that means they've told you 0.43 equals the distance of the spot over solvent front. What you can do is if you do 0.43 times the solvent front, you'll get the distance of the spot. Now, if I try measuring this now with this jank-ass ruler, let's see what we end up getting, eh? So then here, it's what? From 1, whatever mysterious unit this is, right? So 1, 2, 2.6 of this ruler unit, all right? So that means 0.43 times the solvent front, which was 2.6 of these mysterious ruler, is 1.118, right? So that means the distance of the spot is going to be 1.118 of this mysterious thing. So if I go here, that's 1.11234 The closest thing that's that 1.1 really is Z, because that's way higher up, so it's got to be Z. So that's how you do it. You'd obviously measure with your ruler and you get a far more accurate result, but I'm using this and it seemed to have worked. Anyway, thank you very much. I hope that helped. Please like, share, and subscribe, and you know the drill.